What's up, guys? It's 5 o'clock in the morning in Bangladesh, so very good morning to you and welcome to another video from Senda Pharmacist channel. In fact, today I want to talk about something related to the antimicrobial agents. So let's begin with the title, Antimicrobial Agents. So in fact, it's a very big thing. In fact, it covers a whole lot of syllabus, but we don't want to cover that much because if we talk about the syllabus for the PEBC, that is the Pharmacist Evaluating Examination Issues, they just added antibiotics in couple of pages. If I talk about, if you go to the page number 14, you will find certain titles like this: antimicrobial agents, beta-lactams, quinolones, macrolides, clindamycin, tetracycline, aminoglycoside, vancomycin, sulfonamides, dimethoprim, metronidazole. Then they went to antiviral agents, then antifungal agents, but specifically they just mentioned by class only couple of antibiotics from beta-lactam to metronidazole, page number 14. In the same way, they have also added these things in another chapter of this syllabus, that is maybe under pharmacology. Yeah, that is page number 31. They have written anti-infective agents. So in that class they have written antibacterial drugs, antiviral, antifungal, antiprotozoal and anthelmintic. So antibacterial, they have written beta-lactam, carbapenem, sulfonamides, trimethoprim, tetracycline, aminoglycoside, macrolide, fluoroquinolone, vancomycin, metronidazole, nitrofurantoin. So the basic thing that I want to actually share is that although antibiotics or antimicrobial agents is a very big chapter, we don't want to cover that much. Only a specific thing we can cover for preparing ourselves for the PEBC. So those covered area will be beta-lactam, quinolone, macrolide, the basic things that I have already talk to you. So even before going into those things, I think there are certain basics we should at least consider, like antimicrobial agents. If we talk about, we have to know the definition first, and also need to know the classifications, and also need to know a couple of other things, like I have also noted down something, the problems associated with the problems with antibiotics, and obviously we need to know something from the resistance purpose, like the factors affecting the choice of antimicrobial. I mean, based on what factors we will finalize the antibiotics for a particular disease, combination and the purpose of combination, and prophylactic use of antibiotics. So not much things we are going to cover for a basic class, like definition, classification, problems, factors for choosing antibiotic, combination and its purpose, and the prophylactic use of antibiotics. So first of all, if we start with the definition of antimicrobial agents, it is very simple. So the definition, if we talk about, so antimicrobial agents or antibiotic, so it means the drugs those actually either kill microbes, not specifically bacteria, microbes, or retard the growth. So these are the agents that actually kill the microbes or retard the growth of microbes, and these are the substances produced from the microbes. So they are produced from the microbes, and now their works against the microbes, either they kill them or they retard their growth. And while we are talking about killing microbes or retardation of microbial growth, it's the question that either it will kill us or anyhow kill ourselves or anyhow retard our cellular function or not. Obviously, by definition, antibiotics cannot be providing any harmful effect to our cells. So by definition, they have written that little, no harm, or little harm to human. So these are the drugs, those are produced from the microbes itself and used for killing them or retarding their growth without affecting the human significantly, or no harm or little harm. So these are the basic definition of antimicrobial agents. So if we want to classify antimicrobial agents, there are a lot of ways you can classify them. So like you can also classify them based on their chemical structure, also based on their function, based on their activity. There are a lot of ways you can classify them. Like if I tell you what K.D. Tripathi says, based on chemical structure, based on mode of action, based on type of organisms against which primarily they are active, then spectrum, then type of action, then the source of antibiotics. So there are a lot of ways you can classify these antibiotics, but the basic thing is that based on mode of action, this is one funda, and second most important funda is based on chemical structure. So we will talk about this first, based on mode of action. So whatever the antibiotic, I mean, although it is a very big category of drugs, you can classify them based on mode of action only to 8 class. So what they do, number one, there are some antibiotics, those works directly onto the cell wall synthesis. So what can they do? What they can do, number one, inhibit cell wall synthesis. So they are the antibiotics, they directly inhibit the cell wall synthesis. And second thing they can do, they can make holes to the cell wall, I mean perforate cell wall. So on the cell wall they can do two things: either they inhibit the cell wall synthesis, or they can directly make leakage to the cell wall so that the organelles or the inside materials will go outside, causing the cell to death. So these are the two things they can do at the cell wall level. Third thing is that they can interfere with protein synthesis. I mean, they work on the ribosome now. So what they can do, inhibit protein synthesis. Or what they can do, they can facilitate misreading of mRNA. Basically, what do we know about the protein synthesis? The protein is being synthesized by the ribosome. In fact, with the presence of mRNA, they just do the protein synthesis. I mean, this is the basic funda. mRNA is there, I mean, transported to the ribosome. The ribosome actually read the manual from the mRNA and produce the protein accordingly. So what they do, they block the ribosome. I mean, not the whole ribosome, particularly their subunits. They block the subunits and thereby interfere with the protein synthesis. Directly, in fact, they block the protein synthesis. And fourth category is that they can, I mean, the ribosome will be able to read the mRNA, but that reading is imperfect. I mean, they will read wrongly. So ultimately, the wrong protein will be synthesized. So interfere, or I mean, what they can do, misread mRNA, so ultimately causing wrong protein. So if the protein is wrong, ultimately they will not be able to do anything. I mean, what they need from the protein in fact, microbes is a very small thing, so the protein they are having is very essential. I mean, it may be different kinds of enzyme, or different kinds of parameters they need to interfere with the human. So if they don't have any specific protein or specific enzymes, they will not be able to survive. So ultimately they are strongly related with the protein. So if we can interfere with the protein synthesis, or we can trick them by reading the mRNA in a wrong way, they will not be able to survive. The fifth thing is that it is directly related with the DNA level. So what they can do, they just inhibit the DNA gyrase, inhibit DNA gyrase. Somebody call it gyrase, somebody call it gyrase, whatever it is, GYRASE. So I call it gyrase, DNA gyrase. They inhibit the DNA gyrase, and sometimes they inhibit DNA synthesis. So this class is actually antiviral. And number 7, what they can do, they can interfere with DNA function. DNA function. So I think the 3 things they can do at the DNA level, interfere with the DNA function, then DNA synthesis and inhibition of the DNA gyrase. And the last one is that they block intermediary metabolism. I mean, in between they may have different kinds of biochemical process or reaction where these antibiotics or these agents can play a role. So blocks intermediate metabolism. So these are the 8 class of antibiotics based on the mode of action. So if we talk about the antibiotics that inhibit the cell wall synthesis, who are they? Obviously we know from our reading, I mean from our knowledge on pharmacy, we can tell cephalosporin and penicillins, they actually can block the cell wall synthesis. Penicillins, cephalosporins, they can block the cell wall synthesis. Now who can penetrate or who can make holes into cell wall? So these are polymyxin A, amphotericin B, polymyxin, amphotericin B. These are the drugs that can make hole into the cell wall. So cephalosporin, penicillin, they can inhibit the cell wall synthesis. Polymyxin, amphotericin B, they can make holes into the cell wall so that the internal metals will be leaked out. And regarding the protein synthesis, the protein synthesis inhibitor, these are very much popular right now. So it comes azithromycin first. We know azith for a long period of time, azithromycin. Then the drugs which can misread mRNA and inhibit, I mean it's a new concept. I mean the concept is that the ribosome will read the mRNA but they will produce the wrong protein. So it means that they read RNA but wrongly read. So misread mRNA, so that's the wrong protein synthesis happen. So this class includes which antibiotic? These are aminoglycosides. So aminoglycosides again means azithromycins or streptomycins, certain mycins like streptomycins, and inhibit DNA gyrase. This is also another popular class. I mean these 2 classes are very popular, not actually 2, 3. So inhibit DNA gyrase, that class covers mainly the fluoroquinolones. Fluoroquinolones means we talk about ciprofloxacin, levofloxacin, then moxifloxacin, all floxacin type things. Fluoroquinolone, they actually inhibit DNA gyrase or those specific enzyme at the DNA level. And if we talk about this class, inhibit the DNA synthesis, this is, you know, already I have told, antiviral. In fact, they can directly inhibit the DNA synthesis. So it covers, as an antiviral, this covers like stavudine, lamivudine, zidavudine, acyclovir, ganciclovir, penciclovir. A lot of drugs are there in that class. Now the seventh one, interfere with DNA function. It's kind of tricky sentence. So DNA function interference. So it comes metronidazole. Metronidazole. Although metronidazole, it is kind of an imidazole antibiotic that is used for the fungal issues, not actually the fungal things, protozoal issues. So if there is any protozoal infection like you know, dysentery is there, so metronidazole will be there. But other than that, metronidazole itself can interfere with the DNA function, so they can also be part of this category. And the last one is that blocks intermediate metabolism. These are quite interesting concept. I mean, without interfering at the DNA level, or without rupturing the cell wall issues or inhibiting the cell wall synthesis, what they do, they block the intermediate process. So the chemical or biochemical reactions that happens in between, the drugs can actually block those process. So in this category, which one comes? Sulfonamides mainly, I think. Yeah, sulfonamides like timethoprim, then sulfamethoxazole. Yeah, mainly the sulfones and para-aminosalicylic. Those things actually block. Para-aminosalicylic acid, sulfonamides. These are the drugs that block actually the intermediate metabolic process. So this is the classification of antibiotic based on the mode of actions. So now if we talk about the other way of classifying themselves, definitely the second most important will be the chemical classification. I mean the classification based on the chemical structure. So it is, you know, kind of a huge thing. I mean, there are a lot of a lot of chemical structures, so classification will be a lot long list based on the chemical structure. But basically what we can talk about, like, it is not necessarily always that you will classify themselves based on the chemical structures, but at least you need to know the name of those chemical things, like beta-lactams. Beta-lactam antibiotics. This is a very big class, so it covers both cephalosporin and penicillin group. So whatever that fall into that penicillin or penicillin or cephalosporin comes under this umbrella, beta-lactam antibiotics, because they have a beta-lactam ring in their chemical structure. The second most important thing comes as quinolones. So it covers basically the fluoroquinolones, FQ series. So it covers ciprofloxacin, ofloxacin, gadifloxacin, all floxacin things like moxifloxacin. So this is another very important class. Next one is macrolides. These are also very common nowadays, like azithromycin, streptomycin. In fact, they also fall into another big class which we called aminoglycosides. So under aminoglycosides, they fall like macrolides, and the examples are like we have already told. Now there are many other antibiotics like imidazole antibiotics. This is another important, imidazole. So it covers basically the antifungal agents or something like that. Miconazole, then iconazole, metronidazole. These things are imidazole antibiotics. So basically beta-lactam, quinolone, aminoglycoside or macrolides, imidazole, and many other classifications like tetracyclines, then polypeptides, nitrofuran derivatives. These are the basically chemical class. So based on that we can classify them in various ways. So you just need to know a couple of things like beta-lactam, which one is beta-lactam, quinolone, which one is quinolone, which one is macrolide or aminoglycoside, and which one is under this imidazole category. So this is how we can classify them based on the chemical structure. Another way we can classify them, either their broad spectrum activity or short spectrum. Broad and short or narrow, what do we call? So broad means they can cover a wide range of microbes. I mean specifically gram positive and negative, both they can cover. And short spectrum means either they cover only plus or minus. I mean they cover either the gram positive or the gram negative. They are short spectrum or narrow spectrum. If they cover both, they are then broad spectrum antibiotics. But in fact nothing we can conclude about the broad and short spectrum antibiotics, because nowadays from each category, like if we talk about the cephalosporin group, they have a wide range of, we cannot consider cephalosporin into a specific one. I mean you cannot tell that cephalosporin is gonna be a broad or short, because they have all things. Like from the cephalosporin you will find a broad spectrum one, not actually broad spectrum one, couple of broad spectrum antibiotics under the cephalosporin group, and also find certain narrow spectrum antibiotics within the same cephalosporin group. So nowadays it is just a common information, either it is a broad one or narrow one, but we do not use them to classify antibiotics that much. Now another way we can classify them based on the action, I mean type of action. Either they are static, I mean bacteriostatic, or bactericidal. So either they retard the growth of the microbes or they kill the microbes. Based on that we can classify them into these two categories. So if we talk about the static, I mean bacteriostatic, they actually cause the growth retardation of microbes, and within this class, basically if we talk about the bacteriostatic group, there are a lot of things that covers into this thing. Sulfonamides, tetracycline, a lot of drugs are actually used as bacteriostatic. Mainly we can talk about the tetracyclines and sulfonamides within this group. And bactericidal, we have a vast syllabus. So bacteriostatic, we can talk about tetracyclines, and which one? Sulfonamides. Yep, sulfonamides. So tetracycline and sulfonamides, they are bacteriostatic. And bactericidal, we have a huge syllabus like penicillins, cephalosporins. Then I mean the way we can kill the microbes, either at the DNA level, like DNA synthesis inhibitors, or DNA gyrase inhibitors, whoever. So it covers penicillins, cephalosporins, fluoroquinolones, macrolides. Lot of antibiotics are there who can play the role as a cidal agents, but as a static, bacteriostatic, couple of examples are there. Tetracyclines and sulfonamides are the most common amongst. Now we can classify antibiotics or antimicrobial agents in another way. I mean the target microbes. Based on the target microbes we can also classify them, like we can also call them antibacterial. So these are the antimicrobial agents that fight against the bacteria. Then antiviral, fight against the virus. Antifungal, the fight with the fungus. And obviously there are other things like antiprotozoal and anthelmintic. Antiprotozoal and anthelmintic. So antiprotozoal means protozoal infection will be there, and these are the agents which will fight against the protozoa, like dysentery we are talking about, amoebiasis we are talking about, and anthelmintic is basically the albendazole, mebendazole, those things, those actually fight against the worm. So worm infestation if we talk about, then the anthelmintic, antihelminthic or anthelmintic comes there. So these are also the way we can classify the antimicrobial agents. So is there anything pending? Chemical classifications we have done. We have also talked about the classification based on the mode of action, also based on the type of action, spectrum, then target one, and finally one thing we can talk about, like the antibiotics obtained from. So we can classify antibiotics based on the source, how they have been produced. So antibiotics nowadays they are being synthesized commercially, but at the beginning they have been derived from different different microbes itself. The microbes produced the antibiotics, and now they are being used to kill the microbes. So if we talk about the example, so we can derive certain microbes from the fungus or fungi. So this covers the penicillin. Penicillin series actually produced from the fungus. Penicillin, then cephalosporin, these are produced from the fungus. And from bacteria we have received polymyxin series, then bacitracin, those things. And another class is that bacitracin. Another class is actinomycetes. Actinomycetes. It's an interesting class. It's kind of a very specified bacteria who can form the myceli, kind of fungus. Fungus, they can produce a network of them. In the same way, the actinomycetes are kind of a bacteria that can produce myceli or the network. So these are, it's a very big class, and they can contribute in producing antibiotics. Like if we talk about the macrolides, basically the aminoglycosides, macrolides, aminoglycoside, the blockbuster drugs nowadays, azithromycin, macrolides group, then aminoglycosides group, they are being produced by the actinomycetes. So fungus, we get the penicillin, cephalosporin, and bacteria, from bacteria we get polymyxin, bacitracin, etc. And from actinomycetes we get macrolide, aminoglycosides, and many other things. So based on the source we can also classify them. But nowadays the thing is that there are a lot of other ways we can get the antibiotics other than these natural sources. Initially they have been produced by these things naturally, and now they can be synthesized artificially in the lab itself. So I think it's enough for today. Maybe in the next class we'll talk about the factors related for the selection of antibiotics, and also some other issues we need to discuss, like the problems associated with antimicrobial agents use, like the resistance issues, the cross resistance issues, toxicity issues, certain things are there. So hope to see you in the next class. Till then, take care. Goodbye.